Thank you, Steve, and thank you everybody for coming to the whole day today. I think it'll be very exciting to see all these different takes on what is going on in synthetic biology. So for my talk today, our research group, I just want to give an idea of what we do. In practice, we have a group, half the people working on wet lab synthetic biology, the idea being here we're engineering new functions into living cells. And the other half is working in control, algorithms, robotics, here in this picture, kind of wrapping around cells to think of new ways of doing experiments, new ways of engineering cells. And so today I'm going to talk through just one specific area we're working in under this broad philosophy. And as a preview of what I'm going to talk about, I'm going to start with a motivating example of why we're interested in some of the tools I'll talk about. I'm going to then talk about a sticky problem which has arose in the past. I'll talk about some mathematical insights we've worked on to try and get to the bottom of this problem, and how these have then led us to robotic solutions, and finally some emerging biological applications of these. So hopefully in doing this, I can tie together this math, robotics, and biology aspect and give you a good flavor of what it is we're doing. So to begin with, let's think about what common work in synthetic biology might look like. And this is an engineering field. Often we start with thinking of a challenge which we would like to address, medical, maybe environmental, whatever it may be. And so we would go into the lab and we would make one solution and it would work first time every single time. No, unfortunately not. As I think we have some people from our group here will know, I often make that joke, and it's always a disaster. It always leads to partial success, or maybe no success, but hopefully partial success. Let's say we've been lucky enough to get to partial success. What we might do next is try and evolve what we've done towards actually something which is useful. Another approach I might take might be not to be quite so heuristic as this and make one solution, but make a large number of solutions because I accept that I'm bad at what I'm doing and I don't really know which one will work, and then do some kind of test to pick the best out of those. And then I might again evolve it to try and make it more effective. Or maybe I would even just go and look in nature, so what is already out there in nature, which is approximately solving my solution, and take that thing from nature and then try and evolve that towards effectiveness. And so synthetic biology involves all of this, but given that we have a whole bunch of talks today focusing perhaps on all these other aspects of how we do the synthetic biology, today I wanted to talk about some of these evolutionary methods which we're working on our team to try and allow us to improve the performance of some of these synthetic biological systems. So to start then with a motivating example of what we might do, so an example from our lab in the past couple years, Dr. John Allen worked on engineering bacteria here, an electron micrograph we took in one of the facilities in Oxford. You can see these bacteria, and we've programmed them to produce different plastic nanoparticles in different sizes. So depending on the DNA we put into the bacteria, they can produce different granules. You see on the left, they're much bigger than the ones on the right. And we can even go and then program different bacteria to interact with one another and then put some functions on those plastics. We can make them sense different things or bring different enzymes together to more efficiently catalyze reactions. So you can see a cartoon of what this might look like on the bottom left, and then a diagram, some bacteria in a test tube there where we've taken away the cells and we just have these functionalized plastic particles. And so this is all well and good, but actually we might like to make this more efficient. Maybe I'd like to do this in a very sustainable way using some kind of waste product. And an example of this, some work actually by a past DPhil student, Simona, also working with Wei Huang and Ian Thompson in the department, was working on trying to grow some of these bacteria on renewable food sources. So we could imagine building useful things from waste products, kind of doing some circularization there. And so this diagram here we have on the x-axis, we have generations, which is the generations of growth for our cells. So it's about 200 generations of growth from our cells, our bacteria. On the y-axis is the growth rate. And what we're doing in this experiment is trying to convince these cells to better evolve, to better adapt, to grow on some food that they don't really like. It's like we have a child that doesn't like broccoli, and we're just constantly feeding them broccoli until they become big and strong, and now they love broccoli, and they've gotten better. So what we'd like to see in this diagram would be this line, the blue line goes up, which would imply that our cells are getting better and better at growing on this food. And you can see it's kind of doing this, but there's also problems. I mean, you can see that at certain points, these black vertical lines, we put in some ultraviolet radiation to encourage mutations to appear. And there's various other things we'll talk about in a second. But the kind of moral of this is we can kind of get some cells to get better at something we care about, but it's a bit of an up and down. There's some chaos here. They're not just getting better over time. So let's think about what's actually happening in this experiment. So The ideal of what would happen in this experiment is I would have some kind of container here. So shown in my diagram, I have in this container some bacteria floating around. I'm feeding them with some food, so it's the broccoli they don't really like, and I'm taking out the waste in the cells. And so what should happen is I would keep feeding them the food, and they would slowly get better at eating that food, and by the end, they'd be really good. And that would be in an ideal world. However, it's not an ideal world, and this kind of evolutionary assay, which is trying to select cells which do better at what we want, can fail for all kinds of reasons. And one of the most frustrating that we've often come across in our lab is the cells figure out a sneaky way of defeating this system, which is stick to the walls, which is, it sounds very simple. I mean, if you stick to the walls, you'll never get taken out, so you'll just propagate and propagate. But it turns out to be quite difficult to prevent cells sticking to the walls, because basically it's giving them almost like an infinite lifetime because they can't get taken out of the reactor. So there's an incredibly strong benefit if they can just figure out how to stick to the walls. And so Ting in our lab has tried all kinds of stuff to defeat this, feeding them different things, coating test tubes in different coatings like rain repellent. Often people have given us these kind of housewife remedies and they've actually made it worse. Genetically engineering the bacteria to try and remove some of the genes which help them stick also doesn't work. They quickly overcome that. And so basically we've decided that we can't necessarily solve these problems easily unless we have some more complicated methods. And so one thing we can say from this is, well, the cells sticking to the walls is a problem from a practical perspective in that it messes up our experiments, but it's also a fundamental problem in our experiments in that in this case, if I'm trying to convince my cells to grow on the food that they don't like, but actually all my cells do is just learn to stick to the walls, it's fundamentally preventing the evolution that I want. And so this is where we're going to go next, trying to understand why that is happening and how we can maybe get to the bottom of how quickly that will happen. So this was our problems, and this took us to actually the field of mathematical evolution. So this is a kind of science field. We have lots of experts at Oxford here in the biology and biochemistry department working on this, where people are trying to understand how it is that evolution actually happens in these microbial populations. And so this diagram here shows on the x-axis the generation. So this is time passing, and basically what is happening is, you know, at this point here, a new cell appears which is slightly better than its neighbors. This slowly takes over the whole population of cells, so the purple line takes over, and then another cell takes over the blue cell, and so on, and basically every now and then a better cell turns up and takes over the population, and a better and a better. And so slowly over time we see it improve. This is what we might think evolution is doing in practice. It's more like this in these kind of experiments where we actually have a lot of competition between different cells. So I have, you know, millions, billions of cells in this test tube, and they're all competing to make innovations and get better at what it is that they want to be doing to survive, sticking to the walls or otherwise. And so this is a process broadly termed as clonal interference, and kindly the mathematical biologists have done a whole lot of statistical work to understand different ways of modeling this, and eventually come out with some relatively nice equations like this one here, which says the adaptive velocity v, that's basically how quickly the cells get better at whatever it is they're trying to get better at. How long does that take? Well, it's some function of how powerful the mutations are. It's some function of the population size, and it's some function of the mutation rate. And that's when we're assuming that they're all just nicely mixed together and the population is evolving. Our situation, though, is a bit different because we have all these competing problems, like they're trying to get better at eating one food, and they're also trying to stick to the walls. And so this is our kind of situation. We actually have something we care about. So that's my trait 1. That's my goal. That could be recycling the product I would like to recycle. And they could also get distracted. Again, it's like these toddlers running around getting distracted by nicer food than broccoli, and so they don't really do what they're meant to do. So that causes problems for us. And so we can think about what's happening here. We can imagine that to begin with, maybe I have all of my cells are one certain kind, my ancestor cell, and then something mutates to become a little bit better. So my green cell, this is the size of my population, it's slowly growing. So this is like a small number of green cells slowly taking over. Maybe some more good green cells turn up, but then some guy that's really good at sticking to the walls turns up and basically completely annihilates all the other cells because he's so much better at surviving in that environment than the other cells. So that's basically the problem we're seeing here with the experiments. And so working together with people in our team, we spent a lot of time trying to get to the bottom of this mathematically and making some horrendously complicated formulas which tell us how these two traits interact. So here we don't need to worry about the details, but we have some kind of control parameters that we can set in our experiment. We have a similar formula to before, which is telling us how a specific trait is interacting with itself. And then we have this middle term, which basically says, how does the fact that my cells now competing, they have a split attention, how does that mess up the engineering of these cells with evolution? And so we can think about what this looks like. Imagine here my plot is the velocity of adaptation on the y-axis. On the x-axis is how good are my trait 2 mutations. So this is how good are they at sticking to the wall. We can see that if I make those mutations better, well, the red line becomes faster at adapting, which is not surprising. That's basically just saying that over time, if I have larger mutations, I can adapt faster because they're more beneficial. And trait 1 is kind of staying constant because I'm not changing anything to do with trait 1. So this will be the case if I only considered the intra-trait competition, but with our new kind of mathematical formalism here, we can actually see how they would interact. And it's a different story. Actually, what happens is as trait 2 gets more and more potent, it actively stops the first trait from ever happening. So it's saying this thing on the left-hand side is not just happening sometimes, it's guaranteed to happen, and basically will completely prevent what you desire to evolve from ever evolving, because it's constantly being outcompeted by better cells which are cheating our experiment in this way. And so we can see now, like quantitatively, how these things could begin to cause us problems, because we simply cannot prevent them from easily doing this trait 2, and they're always going to cheat in that way, get distracted, and do the wrong thing. So as a recap of what I've said so far, I've said that we have these existing technological approaches, growing cells in bioreactors like this, but they have a whole bunch of problems. We can't necessarily make them evolve as we'd like to improve our technologies. And these are giving us practical problems in that they make it difficult to do experiments, but they're also giving us fundamental mathematical problems where we can show that the traits we care about will simply not happen because of statistics saying that there's just too many beneficial ways to cheat, and so the cells will always take the cheating path. And so this is where robotics come in. We try and build solutions which can actually fight against this. And so our big problem of the cells sticking to the glass is something we can actually overcome with some very clever cleaning systems where we actually have now pairs of reactors which are self-cleaning. And so we can see on the left here, I start out with my cells growing on one test tube here, and I'm cleaning the other side, and then I move my cells across and I clean the first side. And so I go back and forth to try and build a system which is able to prevent some of these pressures. And there's a whole bunch of other pressures that we're trying to eliminate in this case as well, which I won't get into. But the important takeaway is we can then completely delete the ability to stick to the walls as a way of cheating our experiments and making things go in the direction we'd like. And so this is our goal. It's not so easy to do in practice. Basically, the requirements of building such a system are a long way outside of any kind of instrument you can buy off the shelf. And so that's why we spend a lot of time building instruments ourselves to try and solve these problems, which we can't really tackle in any other way. This has a range of challenges, things like reliability, because I have a very complicated instrument. There's all kinds of things moving and measuring, and I'm trying to evolve these cells for months or years at a time. It basically has to run 24/7 with 0 things breaking, and so we have to have all kinds of health monitoring. Everything must be super reliable to allow it to keep doing that over time. I have lots of materials problems, so we're doing really aggressive bleach and ethanol cleaning of every part of the system every couple of hours, which means that all these things like normal plastics and stainless steel are out. We basically have to use tantalum, titanium, a couple of silicon plastics, and PEEK and glass, because anything else will just quickly get degraded over the course of all this really aggressive cleaning to defeat the cells. And we have to then build all the electronics from scratch, because none of this really exists. We have to make sensors which basically have 0 drift over the course of a year. They have very, very precise measurements. And so we have to think about how we can make the best kind of electrical designs to be both supremely reliable, and we can never go in and recalibrate because we need to understand how many cells there are at very low densities of cells. So we have to do a whole lot of interdisciplinary work to make this happen. It then looks like a big Rube Goldberg machine. We have all kinds of stuff happening. We're weighing amounts of liquid. Here's a video on the right. We can see we're putting liquid into these little test tubes on the bottom. We're weighing different amounts of liquid at the top and coming down. We're constantly measuring what the cells are doing in each side. It becomes very, very messy and a very big challenge to program this. And Marco in our lab has done a fantastic job of building all the software behind this in the last couple of years. And so now we're at the point where we can actually very happily grow cells basically indefinitely in these machines and have them really nicely controlled in how they're evolving and understand exactly how quickly this is happening. As examples, we've been collaborating with a group in Harvard where we shipped them one of our machines we built before, And so there in the last year, we've put Vibrio nitrogens, which is the fastest known growing organism, through about 30,000 generations of evolution, which is over 1 million years in terms of human timescale. And so we've now made the even faster organism known to science. And we should be next year catching up with the Lenski experiment, which anyone that's come across this is the longest term experiment in evolutionary biology. It's basically been people manually moving cells around for the last 40 odd years since 1988, and we should have overtaken them next year by using all this automation to allow us to kind of crack down on all these problems. So that's good. Why do we care? Where are we going with this science next? I mean, we're not just doing this to make fun robots. I mean, the first thing we want to do from a scientific perspective is that now that we have the experimental technology and some of the theoretical foundations we need, we can kick off this field we're excited about, which is really thinking about the control and optimal control of microbial evolution. I mean, this is not in science where we might go out and watch what has already happened. This is how in a lab can we optimally control how these processes happen and think about where they would go for a synthetic biological standpoint. And so there we have a whole bunch of different applications. So some examples like the strain engineering I mentioned earlier, working with teams in earth sciences in Oxford to use some of these things in mining and waste reduction. And the one I wanted to very quickly talk about for the last couple minutes are biomedical applications, including antimicrobial resistance, where again we have to understand precisely how cells can evolve in long periods of time in a controlled manner. And so for AMR, we've been lucky to be part of this big US collaboration recently funded by ARPA-H, the Advanced Research Projects Agency for Health, where they funded $100 million in September last year, and we're the international partner on this to try and develop some of these evolutionary technologies to apply them to AMR to understand how we could then predict how cells in a clinic might evolve. So the idea would be here that somebody might be very unwell in a clinic. We might take a sample of the bacteria which is resistant, for example, in their blood, and then try and fast forward this adaptation to see how might it respond to different antibiotic treatments, which then tells our doctor or clinician how they should actually allocate these medicines. This is very much not easy. We don't really know if we can do this perfectly, but we think we're going in a good path where we know that we can't perfectly replicate the environment inside a human in a lab. It's just never going to happen. So we're trying to think about how can we just make it as sensibly similar as possible using some of these theories where we can show kind of mathematical equivalences. And so we now have the theory, we have the technology to test these ideas. So what are we doing? We are first tackling a whole bunch of technical challenges. I mean, it might sound easy on the first hand, but actually we need to make these systems even better and more stable if we're going to use them in a clinic. For example, because if we're going to take samples out of a person and make them even more antibiotic resistant, we need to have very, very close control of what happens to those cells because they're going to be even more dangerous than they were in the person. And so we're thankfully not doing this here in the department. Don't worry, Ron. We're shipping the machines once we've got them working here to hospitals in Boston where they can actually do these tests in a proper controlled lab environment. So we're not responsible for the dangerous clinical samples. And so some of these are the technical challenges, and some of the things I'm very excited about from the theory and biology is thinking how can we actually build this into a field more like engineering where we're thinking about some of these formulas I showed earlier and trying to make things like characteristic parameters for evolution. So think the Reynolds number for fluid dynamics. We know that in engineering we can make scaled experiments with a similar Reynolds number to somehow get similar results. How could I define such numbers for biological evolution? And then how could I actually implement experiments which I know are not real life, in that I know a 1 to 10 scale model in a wind tunnel is not a perfect explanation of how a plane would behave in the air, but maybe I can say that it's got the same statistical properties, which are the things that I care about in my specific applications. So we're really trying to take this engineering approach to think about where we can send this field in the future. So I think I'm exactly at time, so I better sum up. So I hopefully have convinced you of the following, that I started off with an example applied to bacteria accumulating some plastic that we'd programmed, and that this was actually not so easy in many cases to evolve them to become better because basically the cells would stick to the glass in my test tube and cause me all kinds of problems. And that was not just a problem from implementing an experiment. That was a fundamental problem we could show mathematically, was basically going to destroy my experiment and we could prove that it would never actually allow you to evolve the thing you want as you would want. But hopefully we can solve that with robotics, and then we can hopefully apply this to all these different challenges we're working on now in antibiotic resistance. And as a final summary, if this does not look like something you can remember tomorrow, you can alternatively imagine my talk as us being a bunch of kind of demons trying to intervene in this romance between bacteria and glass, which has been going for all time in all kinds of areas. And the bacteria just want to stick to the glass. And we're just trying to come up with technologies to prevent them doing that so we can get the data and experiments done that we actually need. So with that, thank you very much, and a special thank you to our team and funders.